Thank you everybody for joining online and in person for the first session of this year's WHRI Summer Scholars Series event. And we begin by acknowledging that the WHRI operates on the traditional, ancestral, and unceded territory of the Coast Salish peoples, Musqueam, Squamish, and Tsleil-Waututh nations. Further, this acknowledgement, gratitude, and respect extends to all the First Nations communities on whose traditional territories WHRI builds relationships. 2025 marks our third annual Summer Scholars Series event, with this year's theme being Sex and Gender-Based Analysis Plus across the health research pillars. We are pleased to welcome Dr. Julian Christians as our opening speaker. Dr. Christians is professor and chair of physiology and genetics at Simon Fraser University. He joined SFU in 2006 following a PhD in ecological physiology and postdoctoral work in quantitative genetics. His research interests include fetal and placental responses to suboptimal environments, how prenatal environment affects health in adulthood, and the ways in which responses and long-term effects differ between the sexes. More recently, Dr. Christians has explored the potential use of epigenetic markers to assess gendered exposures to distinguish effects of sex and gender in health research. And he served on the review panel for the CIHR Gender Sex Health Committee from 2022 to 2025. So before we begin, a few technical issues for in-person attendance. Press the button on your microphone prior to asking questions at the Q&A. For online participants, you may submit your questions via the chat, and Olivia Patrick will ensure they are relayed to Dr. Christians. We also invite you to the next two sessions, first July 9th with Dr. Rachel Olivier on integrating sex and gender considerations in cardiovascular care in Canada. And then August 13th with the Indigenous Gender-Based Analysis Plus team, who will present on Indigenous Gender-Based Analysis Plus. Registration for these events are available through Eventbrite, accessible via WHRI's website and social media. And then concluding this event series, we will be circulating an evaluation form in August and would greatly appreciate everyone's feedback. So please join me in welcoming Dr. Julian Christians.
Thanks very much for the invitation. I will hide behind the podium just so that I can use the laser pointer so that everybody online can see the same thing. So I'll first start by talking about what I will or will not be talking about, because this is obviously a big area with lots of different issues. So I'm going to be talking about some robust approaches to assessing whether treatments or exposures differ between, have different effects in males and females. What I won't be covering is the distinction between sex and gender and or the interrelation between their effects. Nicole Rasheen and also Wendy Robinson and I have a project that's looking at that. And so if you're interested in that, I'd love to talk to you. Happy to share bibliography that we're putting together that captures a lot of what's being done in that area. So very important, but not what I'm going to talk about today. Another thing that I won't be doing today is thinking about sex in non-binary ways. So for this talk, I'll just be accepting male and female as categories, which some of the time that will make sense, but some of the time it won't, right? So if we're looking at physiological processes or health outcomes that are, for instance, heavily influenced by gonadal hormones, maybe it makes more sense to actually look at hormone levels rather than just having discrete categories. So again, I just want to flag the limitations of this talk, and that there's a lot more issues even just for considering basic science. Now, at times in this talk, you may feel like I've gone off on a rant, and that may be true. But I want to emphasize that we're probably still on the same page. So I do want to emphasize that we should always consider the potential for sex differences, and that sex differences can be important. But what I'll be ranting about is that just because someone claims that there is a sex difference doesn't necessarily mean that there is one, or that it is either biologically important or clinically important. And I'll provide some examples of that later on. And so this is sort of how I see the field of sex and gender and health research, that for a long time, and hopefully to a diminishing extent, people have just focused on male humans and animal models, and haven't thought about anything different. And that's clearly not a useful approach and harmful in many cases. But I think in some fields, at least with some researchers, the pendulum swung a bit too far to the extent that their sex and gender are expected to always affect everything. And so I think that is not accurate. I think probably the way things are what I've shown in the middle here, is that sex and gender are important and should always be taken into account. And I've deliberately put, where the pendulum is now, a bit closer to reality than where we were before. So to illustrate these points, I will use examples from my own research interests, which, as were noted, are in prenatal health and placental biology. And so these observations are very well established, that pregnancies with a male fetus are at increased risk of preterm birth and stillbirth, other pregnancy complications such as gestational diabetes and term preeclampsia, whereas pregnancies with a female fetus are at an increased risk of preterm preeclampsia. And the associations with preeclampsia are less strong, and certainly the associations with preterm birth and stillbirth are much stronger. And so these observations altogether are often interpreted to mean that male fetuses are more vulnerable. And I'll come back to why that might be in a moment. But this misses an important consideration, which is that sex ratio at conception. The best data we have that I know of is that the sex ratio at conception is 50-50. And overall, the total female mortality during pregnancy exceeds total male mortality. So when studies have looked at rates of preterm birth, rates of stillbirth, they're looking at a selected population, right? Because you're only including in your study those conceptions that have survived to a certain age. And so that's one reason why, while I absolutely don't dispute, there's very strong data that there is this sex difference if a conception gets to 20 weeks, the overall interpretation that males are more vulnerable is a bit more complicated. And there are other things that I begin to disagree with more. And so one of the interpretations of why males might be more vulnerable is this idea that boys live dangerously in the womb. So this was a cunning way to title a paper, a very provocative title. I don't really agree with that. So one of the things they say is that in the womb, boys have a more dangerous growth strategy than girls. They grow more rapidly and invest less in placental growth, which puts them at greater risk of becoming undernourished. So, again, the observation that stillbirth rates and preterm birth rates are higher if a conceptus gets to 20 weeks, not arguing with that. But the idea that that males are prioritizing one thing over another, there just isn't, the data in this paper just doesn't actually support that. This statement obviously mentions placental growth, and the placenta itself is mostly derived from cells of the conceptus. And so the placenta has a sex as well in terms of its sex chromosomal complement. And this is a more recent paper that talks about the effects of placental sex. And they continue this idea that males are more vulnerable because they are investing more in growth. And right, so males are over here, so they are investing more in growth. They have a lower placental reserve capacity, which they don't really describe, but one can imagine having sort of more placenta than you need. Might be a definition of reserve capacity, and that the fetus and the placenta are less adaptable, which again is a little bit vague, but is generally meant that to mean that the fetus and the placenta are less responsive to environmental signals. So the idea is, and really I'm not setting up a strong man here, the idea is that even in the face of adversity, males will just press on and try and grow quickly and grow more, whereas females are the opposite. They're more adaptable, and so will adjust their growth. And as a result of this, females will have increased survival, whereas males will have increased mortality and morbidity. Now, why would males do this seemingly harmful strategy? And again, I'm quoting to show that I'm not just making this up or putting words in their mouth. So they say it's to maximize growth through to adulthood, thereby ensuring the greatest chance of reproductive success. Which makes sense, but why wouldn't females want to do that too? And so this is the closest that I've seen people come to actually saying, you know, boys or males have to be big and strong in order to compete with mates. But I think that's behind some of these ideas. So I use this as an example of how, even though a lot of this work is studying basic research, basic questions about basic functions of how placentas work and how fetuses grow, in many cases in animal models, it's being shaped by, I think, social perceptions of how males and females are in our society. So some challenges to studying sex differences in the effects of, say, prenatal environment or indeed in other contexts, is that it's hard to get a grasp on what's really going on, right? And that's in part due to publication bias. If you find a sex-dependent effect, you'll say, hey, look at this sex-dependent effect. It's really neat. Whereas if you don't, you might still publish the work and publish that there was an effect of prenatal environment on the pregnancy outcome or so on. But if there isn't a sex-dependent effect, you're not going to make a big deal out of it. Another problem is cherry picking the data. So I've described this hypothesis that males invest more in growth and less in placental development, and you can certainly find data that will support that. But you have to sort of ignore a bunch of other data that doesn't support it. And of course, a solution to this is performing systematic reviews. So I imagine this audience is pretty familiar with systematic reviews. So I won't belabor how they work, but just to point out that they often involve registration. So this is a registration of one of ours at Prospero. And so this keeps us honest, right? Because we have to define what the question is that we're asking, what studies we're going to exclude, why we're going to exclude certain studies. And this is all made publicly available before we even do our study. So I'll describe a couple of systematic reviews that we've already published. This one, as the name suggests, it was about sex differences in the effects of prematurity or low birth weight on neurodevelopmental outcomes. So here again, the idea is that if males are prioritizing growth over something else, and if they're less responsive to prenatal challenges, then we expect males to be more affected when things go bad. So for instance, if there's a very low birth weight or if they're born premature. So we looked at studies that examined later neurodevelopmental outcomes. And this is just for one of the meta-analyses we did. So this was for the effect of prematurity on cognition, basically measured through things like IQ and so on. So we have all the studies here are the same as the studies here. And these are their data for males. These are their data for females. And you can see that on average, if an infant was born premature, there was some decrease in their cognitive ability measured later in life. And these are the ages, so quite a range of ages at which the offspring were measured. And so this diamond is the average value for males, this diamond's the average value for females, and this diamond is the difference between the effect of males and females. And you can see that it overlaps with 0, the 95% confidence interval for the difference in the difference between males and females is not significantly different from 0. So the effects of prematurity on cognitive ability in this case were not sex dependent. And we looked at a bunch of different traits. So this is for cognitive traits, but we also looked at internalizing traits, things like anxiety, externalizing traits, things like disruptive behaviors. We looked separately at severe prematurity or severe low birth weight versus moderate. Any which way you slice it, we did not detect any evidence of sex-dependent effects. There were also a bunch of other traits that were hard to combine into a meta-analysis because some data would be presented as means between groups and some would be presented as odds ratios, or they were sort of more obscure measures of cognition or language. So they were harder to sort of fit together into a meta-analysis. And so we summarized the data like this based on whether both sexes were affected, whether females were more affected, whether males were more affected, or whether neither was affected. And we looked at different ages. Not surprisingly, there's more studies of children because they're easier to do. You don't need to follow the participants as long. And what you can see is that there are some studies that, so each of these points is a study and the number refers to the reference in the bibliography. There are a number of studies that find sex-dependent effects, but they're roughly similar numbers for both males and females. And it's much more common to either find both sexes affected or neither sex affected. So when you look systematically at the data, you don't, at least in these, for these outcomes and for these exposures, you don't find that males are more vulnerable. We also did ask similar questions in rodent models. So here we looked at the effects of prenatal food or protein restriction. And again, testing this idea that males are investing less in the placenta and so more vulnerable and less able to respond to a prenatal challenge. So here we looked at studies, and a surprising number of studies have done this, have just manipulated food restriction in the second half of pregnancy, right? So pregnancy's tootling along nicely. Everything's a nice nutritional environment. And then all of a sudden, there's a reduction in either the level of the protein content of what the mother's eating, or the number of calories that the female can consume. And so again, if it's true that males have less reserve and are less able to respond, males should get hit by that more severely. And we did some systematic reviews or meta-analyses of a couple of traits. There weren't that many traits that were comparable across multiple species, multiple studies. But we also, again, summed up traits that were more affected in males, more affected in females, or where they were affected in both sexes. And when you look at all of the studies, it's a little bit all over the place, right? So some studies will find some traits to be more affected in males, but other traits to be more affected in females. So again, not a lot of consistent evidence when you look systematically that males are more affected by adversity prenatally. So another problem, which I think contributes to sort of muddiness in the literature, is inappropriate statistical analysis. And this has to do with type 1 errors versus type 2 errors. Now, if you're like me, you may never be able to get straight which is which. And that's okay, because what's really important is what the errors mean. So when you're doing a statistical analysis, you can make a couple of different kinds of mistakes. I mean, hopefully you get the right answer, but sometimes you're going to make mistakes. And so one of the mistakes you can do is say that there's a difference or say that there's an effect when there really isn't an effect. And so that's why we worry about things like p-values. The other thing you can do is say that there isn't evidence of an effect when there really is an effect. And so when we talk about power analysis and we do sample size calculations, that's what we're worried about. And overall, we're probably more worried about the first one. We think more about p-values than we do about power analysis. So let's think about what those two errors will cause when we try and look for sex differences, and particularly if we do sex stratified analyses, if we analyze males and females separately. So consider this hypothetical scenario where the reality is that the effect of the environment or some exposure or some treatment, whatever you're interested in, just imagine that it's the same in both sexes. And imagine that, given your experimental design, the probability that you're going to detect that real difference is about 0.8. So that would be your statistical power. And that's, I think, considered pretty good. If you can get that power, that's considered pretty good. Okay, so what's going to happen if those two things are true? Well, if you analyze the sexes separately, this is basic probability, the probability of two events both happening. So the probability that you will find the effect to be significant in both males and females is 0.8 times 0.8 is this. The probability that it will be significant in neither sex is 4%. And so the remaining probability is the probability that the effect will be significant in one sex, but not the other. And in this example, that probability is 0.32, which is kind of substantial, right? That you had good statistical power. The reality is that the effect of environment or treatment or whatever was the same in both sexes, but there's a 1 in 3 chance that you're going to say that it's a sex-specific effect. And that's a false sex-specific effect. And people are doing this all the time, right? They're doing sex stratified analyses, and there are some substantial probabilities that they're getting these false sex-specific effects. And I'll give some more examples of that later. Now, a solution to this is to test for statistical interactions. And I use this figure to sort of illustrate what a statistical interaction is, because I've found it's just they're harder to get your head around what a statistical interaction is. So say you have some hypothetical relationship where environment affects phenotype, and of course we're interested in health outcomes. So perhaps some form of stress or some prenatal adversity, we're interested in its effect on disease. So you can see here in both males and females, higher stress increases the risk of disease. And you can also see that males have an increased risk of disease compared to females. So if this was the way the world worked for this particular scenario, there's clearly an effect of sex. Males are at higher risk than females. And there's an effect of environment, that high stress increases the risk of disease. But that effect affects males and females equally, right? So there's no interaction between sex and environment. And that would be in contrast to a scenario like this, where in this case, there's really no difference between males and females in low stress environments. But in this case that I've drawn, males are much more susceptible to the effects of the environment. And females experience an increased risk as well in high stress environments, but the males really get nailed by that environment. So to look at these effects, we tried, we had a publicly available data set where there was all sorts of data about pregnancy outcomes, and then they followed the offspring, human offspring, 7 years later and measured a whole bunch of aspects of cognition. And so we asked, well, does being born small for gestational age, does that affect cognition? Does being born premature, independent of the effects of birth weight, affect cognition? And we also looked at the effects of whether the pregnancy was affected by preeclampsia. So we did the analysis both doing interactions, which is what I'm saying you should do, and also doing sex stratified analyses. So these are the results. I know the text's really small. Don't worry about reading the individual text, but all of these rows are the different cognitive traits that we looked at. And so this table is just for the effects of SGA, small for gestational age. And so these are the results from the combined analyses where we asked, is there an effect of being small for gestational age? Is there an interaction with sex? And then these are the results from the sex stratified analyses, where we analyzed males and females separately. And if you look, you don't need to look closely, I'll just tell you, but most of these interactions are not significant, but there are many cases where the effect is significant in one sex, but not another. And so we refer to these as spurious sex-specific effects. And so wherever there's yes, that's where we've identified a case where the interaction isn't significant, and yet the sex stratified analysis would say that there is a difference between the sexes. And you can see there's only 2 no's and a whole lot of yeses. So now some will say very fairly and accurately that, well, maybe the interaction isn't the right answer, and that and certainly it's true that you have lower statistical power to detect significant interactions, and so maybe the sex stratified analysis is better. And so we've looked at that in a couple of different ways. And so one approach to considering that is to not only perform the analysis separately by sex, but then to compare the effect size in males and females. So to illustrate this, I'll use one of our recent experiments where we studied the effects of food restriction on pregnancy and specifically on placental gene expression. So as I described in the studies that we systematically reviewed before, we let the pregnant female mice in this case, eat however much they wanted for the first half of pregnancy. And then in the second half of pregnancy, we reduced the amount of food available to them to about 60% of what they would have otherwise eaten. And then looked at gene expression in the placentas with RNA sequencing. So again, we're testing this idea that if males have been keeping less in reserve or less able to respond to adversity, we should be able to see that with an experiment like this, where everything's tickety-boo for the first half, and then all of a sudden they're faced with this challenge. Okay, so we looked at the number of genes that were significantly affected by the effects of food restriction in the placenta. So for reference, RNA sequencing will find a whole bunch of genes. So we were looking at about 17,000 genes. And so we did what we thought we should do. We analyzed the sexes together and included an interaction term. And we also did sex stratified analyses. And when we analyzed the sexes together, we found one significant interaction out of 17,000. So this is comparing the number of genes found to be significantly affected by food restriction in the different analyses we did, right? So the yellows, when we combined males and females and included an interaction term. So there was only one interaction term, but there were hundreds of significant genes that were affected by food restriction. Over here, the genes that were significant in XX mice, and over here, the genes that were significant in XY mice. And you can see that there's lots of sex-specific effects, genes that are significant in one sex but not the other. And also there's a whole bunch of genes, like this 428 and the 119, that weren't even significant in the combined analysis. So just looking at this data, or if you had only performed the sex-specific analyses, which is not unusual, you would say there's a lot of sex-specific effects going on here. But now this is what you see if you look at the actual effect sizes estimated in males and females separately. So this is, again, a difference in a difference. So the log fold change is how big an effect there was of food restriction, right? So if there was a log fold change of 1, that would mean that the gene was expressed at twice as much in food restriction than it was in control. And if it was -1, then it would be half as much. And so this is the difference in that effect between XX and XY mice. And so you can see that most of the effects are around 0. So if we just consider this bar here, of the 700 or so genes that were only significant in females and not significant in males, about 200 of them, the effect size is almost exactly the same in males and females. And for most of the others, the effect size is pretty close in males and females. So even though you have loads of sex-specific, in air quotes, genes, the effects are generally very similar in males and females. So if you only relied on this sex-stratified analysis, I think that result would be very misleading. And this is another way to look at the data. This isn't in the paper, but this is plotting for genes that were significant in either the combined analysis or one of the sex stratified analyses. So each point here is a gene, and there was a significant effect of food restriction in some analysis, and the colors are telling you which analysis it was significant in. So if they're gold, it was significant in both. But if it's red, it was only significant in females. If it was blue, it was only significant in males. So you can see that there's a bunch of genes that were sex-specific here. But males and females are pretty much doing the same thing, right? In both sexes, the estimate of the effect size is that they're increasing substantially in both sexes. But then in contrast, there's some genes, like if you look at the red genes along this line, these have very essentially no effect in males, but they have some potentially substantial effects in females. So maybe those are real. They didn't show interactions, and so I would say, don't worry about them, but maybe they're real. So a way to address this is through permutation tests. So when this paper was being reviewed, one of the reviewers said, well, maybe the issue is that you don't have statistical power to detect the interactions, and maybe these effects are actually real. And yeah, maybe a bunch of them are small, but maybe they're biologically important. So they didn't disagree with our analysis, but they thought we should tone it down a bit, and that maybe these were actually real effects. And I was like, well, okay, fair point. But so then I tried doing a permutation test to address that possibility. And so first I'll explain what a permutation test is. So this is roughly what our data looked like, our RNA sequencing data looked like, right? So we have some samples that are male, some samples that are female, some samples that are one treatment, so say food restriction, or B, say controls. And then we've got a bunch of genes, right? So 17,000 genes, I'm just showing 3 here. And then the number of counts, so a measure of their expression levels. And obviously we had a lot more than 7 samples. We had about 60 samples. So I'm just showing you a little snippet to give you a taste of what the data looks like. And you can imagine that if you did some other omics study, you wouldn't have the counts of expression counts, but you'd have some other numbers here. So with a permutation test, you first do an analysis, as we did, on the real data, and you get the numbers that you're interested in. So in this example, we're interested in the number of sex-specific effects of condition, which I showed you in the previous slide was around 700 female-specific effects. Now, in a permutation test, you then permute sex relative to the rest of the data set. So here's what that might look like. So this part is exactly the same as it was before, but you just randomize the sex. So effectively, you destroy any association with sex that there might be. And then you calculate the statistic of interest. So in our case, the number of sex-specific genes. And you do this many times. So depending on how good your computer is, it will take a laptop about 5 hours to do 1,000. And that seems to be okay, pretty standard in the literature. So this will give you a distribution of what your statistic of interest looks like under the null hypothesis that there's no sex differences, right? Because we're just assigning sex at random. But everything else about the data set, the distribution of the number of genes you're analyzing, the distribution of the counts, the effects of condition, the sample sizes per condition, all of that stuff is kept constant. So then you can compare your statistic of interest from your real data to this distribution under the null hypothesis that there's no effects of sex. So what did we find? So that we found more female-specific effects in 375 of the 1,000 permutations, right? So p equals 0.375. So even though we found a lot of female-specific effects in our real data, we found that many or more in a lot of the permutations. And what about these? These little bars here, which indicate a gene where males and females showed really different effects of food restriction. Well, we found as many as are shown here, where the difference in effect size is greater than 1 or less than -1. We found that in the permutations 875 times. So if we go back to this figure and we think about the scatter around, just a straight line, some of that scatter is just due to chance. And so what that analysis said is that, a lot of the time you'll have genes like this, where just due to chance, the effect in males is really different from the effect in females. So one could still say, well, maybe some of these effects are real. Maybe you have low statistical power to detect interactions. Maybe these small effects are biologically important. That's all still true. We can't really address that. But if the evidence of sex-specific effects is not as good as using a random number generator, that's by definition not really good evidence. So, yes, maybe there's something going on, but this data does not demonstrate that. Okay, so I'll end by putting in a plug for a mouse model that I use in my own research, which gets at the mechanisms of sex differences. Right, so very, very simply, sex differences could be due to a difference in chromosomal complement, the difference between having 2 X chromosomes or an X and a Y, and that could be due to the number of X chromosomes you have or the presence of a Y chromosome. Or it could be due to the presence of gonadal hormones. And those might exert effects prenatally or pre-puberty or post-puberty. And so understanding which of these are contributing to differences is important for understanding what I talked about earlier about whether and to what extent it makes sense to just classify and compare males and females versus looking at actually what you think might be contributing to the difference. So there's this neat model called the 4-core genotypes, which consists of a double transgenic mouse. So here we have an XX gonadal female. So this is just a totally normal wild type female. This mouse, this XY minus SRY, so this Y minus indicates that on its Y chromosome, it has a deletion in the SRY gene. The SRY gene is essentially what triggers the development of testes during prenatal development. So that gene doesn't work on their Y chromosome, but they have the SRY gene as a transgene inserted into one of their autosomes, a non-sex chromosome. So this is what happens when these two mice are mated together. Simple Mendelian genetics, the female is going to pass on an X chromosome to all of her offspring, right? So one of these X chromosomes is from the female in all cases. Now the male can pass on the X chromosome or this Y minus chromosome, right? So either the X chromosome or the Y minus. And then he only has one copy of this transgene on one of the autosomes. So that will either be passed on to the offspring or not. So you'll get these 4 genotypes, which are the 4-core genotypes. And so these ones don't have an SRY gene, so they are gonadal females. And these ones do have an SRY gene, so they're gonadal males. So you can get the usual XX females and XY males, but then you also get XY gonadal females and XX gonadal males. So you can study the effects of chromosomes and gonadal status separately. And this model has been used tons in a lot of behavioral research and a lot of cardiovascular research, but not much at all in terms of prenatal development or the long-term effects of prenatal environment on health. And basically, sex chromosomes affect everything. So I think the public perception or the common perception is that aggression is related to testosterone, which is true, but sex chromosomes also influence a whole bunch of different behaviors and other sexually differentiated phenotypes. So gonadal hormones are also important, but chromosomal complement is important as well. So this is the same slide I showed earlier. So yes, we should always be on the lookout for sex differences, and in some cases they are important, but there's also a lot of literature out there, at least in my field, which makes claims of sex differences, which I don't think are well supported. And in particular, I think this issue of performing stratified analyses without following up somehow by looking at effect size or by doing either interactions, or there's other ways to test whether there's a significant moderating effect of sex, will often lead to misleading results. So I'll wrap up there, and I'm happy to take any questions. Yeah.
Let me see if I can use the mic. So I think most of the studies you were talking about, the primary objective is to look at the sex differences. But do you think these problems are magnified in studies where the sex difference isn't the main analyses, but people look at it as a secondary analysis, and the problem of small numbers becomes even more magnified?
Yeah, I mean, it's a bit hard to say what the author's initial motivations were, right? So that's another. So in more clinical studies, obviously you have the primary outcomes and the secondary outcomes, right? But in basic science, we don't do that. And so there's more room for sloppiness, right? Not, like I do it too, right? You talk about what's interesting and where the differences are. So I think a lot of the papers on sex differences have come about because they analyze the data and they analyzed it in a bunch of different ways, and they're like, oh, wow, if we analyze the males and females separately, look at all that happens. And in, like part of it is motivated by wanting to take sex into account, which we should, right? And part of it is responding to that need to consider that males and females might be different. So it's not nefarious. It's and some resources that advocate and emphasize the importance of taking sex into account will recommend stratified analyses. So, yeah, I don't think it's a lot of the literature I encounter is not primarily looking at the sex differences, but it's a belief about, say, how placentas work has been influenced by that thinking.
Here we go. Thank you. It's very interesting to hear all of this and inspiring to then try to apply it on our own research. So I was wondering, we've always used p-values to then basically make decisions, right? If it's significant, we trust something. If it's not significant, we don't think that's true. But it looks like we shouldn't only be relying on p-values. And I wanted maybe to hear more about it. Like, how should we interpret a p-value or the results that we get?
Well, so I think, okay, so there's p-values, which, and I know, how our lives are ruled by p-values, that it's significant or not. But then there's, well, is it biologically important? Is it clinically important? And so that comes to effect size. And so you can look at, for instance, the confidence interval for the difference between males and females. And so maybe that confidence interval includes 0. And so, the interaction wouldn't be significant. And I'd say, no, there's nothing there. But maybe the confidence interval is very large. And so there could be some clinically relevant difference. So that's one answer is to examine effect sizes, like I was talking about, but also the confidence intervals for how big the difference could be. The other thing is to think about what p-values mean, right? So if you do a sex stratified analysis, yes, you get a p-value for males and a p-value for females, but the p-value for males is, can I say that this effect or whatever is significantly different from 0 in males? Can I say that it's significantly different from 0 in females? That's a different question than saying, is the effect in males different from the effect in females? So even if you're going to be ruled by p-values, you have to think about what the question is that you're asking, right? And we would never say, oh, well, the effect of in the treatment group, it was significantly different from 0, but in the control group, it wasn't significantly different from 0. Therefore, the treatment worked. We would never do that, but that's exactly what we do when we do sex stratified analyses. I'll put in a bit more of a plug for the permutation test because just to address that issue that the permutation tests allow you to ask more different questions. So rather than just saying, well, is this gene significant or is it not? You can say, well, how many genes show sex differences? And is that different? How many show big sex differences? So you can tailor your question and make it more biologically relevant or more clinically relevant, and test that with a permutation test in a way that you maybe can't do with just normal parametric statistics and t-tests, right? Like if you say, oh, well, I got 700 sex-specific effects, is that more than I expected to chance, right? Yeah, so the permutation test allows you to answer more specific questions like that. Yeah.
I'll just follow up on that one. So, because when you were showing the permutation test, then it looked like you mentioned it could be by chance. But I was wondering, is it chance or residual confounding that we're not measuring with the current variables that we have? So it basically is the relationship between the outcome and the genes that we're seeing. Is there it could be the hormone levels that we're not measuring and that it's what's driving the differences, or it's basically by chance?
Well, in the case of the permutation tests, if something is, so I was, is that still up there? Right. So the permutation test is breaking the association between sex and the gene expression, right? Like they're no longer associated. So any associations are due to chance. So to whatever extent something is confounded specifically with sex, so, if it's because the females have 2 X chromosomes or because they have lower testosterone, whatever that is, if it's whatever is associated with sex, that will also be, the association will be broken because that's also being permutated. Yeah. Is there anyone?
No questions online.
Okay.
Yeah.
Okay, my pleasure.